the cell in a hypotonic solution water is gonna flow in. Water is gonna flow in. Does that make sense to you? Who says no? Go ahead. It's all right. You said no. Who said it? Someone said no. It doesn't make sense. Is that person brave enough to say who they are so they can explain what does not make sense? No? Okay. So hypotonic. Water's going to go into the cell. Why is water going to go into the cell if it's hypotonic? I'm going to say, I've said it, I don't know how many times. You've hopefully answered this document. You've read it. You've done the Nearpod. So hopefully you've done Nearpod. If you haven't done Nearpod, I would invest in doing the Nearpod. It talks about all of this. So think about this for a second. If it's hypotonic, that means the solute is low. That means the solvent is high. So that means water is high. So if this is hypotonic, then inside must be hypertonic, which means high solute. 2 rules I told you to memorize. I don't care which one you memorize, I told you, because both will get you the right answer. I said water, if water's going to move, which only happens if the solute can't, if water's going to move, then it's going to move towards hyper. So we said, it's a simple rule. So this is hypo, that means the inside must see hyper, water's going to move in. Boom, done. The other option is to think about water's being in high outside and water is low inside. So water moves from its high to its low. Both ways of thinking about it give you water going into the cell. I mean, it's just that simple. And because they're going into the cell, the water swells. The cell swells. It keeps swelling like a water. What happens if you keep filling up a water balloon? It'll burst. I can't tell you how many water balloons I've bursted in my life. And so the cell will burst and water will come flowing out. The thing will die. This is called, in an animal cell, it's called lysed. Lysis. It's a breaking or splitting up or whatever. Now, the cell that they used in the diagram is a red blood cell. So a red blood cell looks like this, remember? We talked about it and the difference between sickle cells. So when it swells, it looks like this. It swells, it looks like this. And then when it bursts, if it keeps swelling, when it bursts, it looks like something like that. All right, so when we're talking, no matter how you look at it, the bottom line is the cell will swell until it bursts because it's in a hypotonic solution. So that's the drawing you have there in the hypotonic solution. When it's isotonic, the cell has its normal shape. But when you put it in a hypertonic solution, this is iso, meaning the same. This is hypo. So when you have hyper, what did we say? What was the rule? What happens to the cell when it's in a hypertonic solution? Where does this cell? So you have this cell, it looks like this normally. Then you put it in a hypertonic solution by adding a lot of salt or sugar or taking away water, however you want to look at it. What's going to happen to the cell? It's not going to swell. Water's not going to go in. Water goes towards the hyper side, which is outside. So the cell's going to shrink. The cell's going to become really small. And because it's going to lose water, water's going to come out of the cell. So we call that plasmolyzed. That's plasmolyzed. A cell that shrinks so bad that it can't work anymore and it dies is called plasmolyzed. That's in a hypertonic solution. That's for animal cells. Now, plant cell, oh wait, I'm sorry. I shouldn't have said plasmolyzed. I lied to you. I apologize. I should say I made a mistake, but anyways, it's called crenated in animal cells. Crenated. So cell, you need to know that word for tomorrow because it's either crenated or plasmolyzed, depending if it's animal or plant. All right, yeah. Which one, darling? The one, which one? So if you take that cell and you put it in a hypertonic solution, in what direction will water move? Remember the rule I told you no, right? I said 3 ways of thinking about osmosis. We're talking about osmosis here. There's 3 ways of thinking about it. Diffusion is easy, right? Simple diffusion is easy. All diffusion is going from high, something's high to something's low, correct? That's diffusion. That's always, you see a diffusion problem, you're like, got it, done. Thank you, Jesus. Right? That's easy. You get osmosis, it gets a little tricky because you have to think a little more. Not much more, but a little more. One more step. The trick to osmosis is water will move not from the solutes high to the solutes low. No. Water will move, you can think of it in 2 ways, towards the hypertonic, or water will, you can think of it another way, water will move from its high to its low. Is that clear? So either way. So here's this cell. I'm going to take it and put it in a hypertonic solution. Where's water going to go? Out. So water's leaving. What's happening to the cell? It's shrinking. That shrinking, we call it crenated. Just a word, crenated. Just means it's shrinking so much that the cell dies. Hypo, it swells so much that it lyses. That's what happens in animal cells. Now it's going to be different. It's going to be different in plant cells because plants have something else. What do the plants have? Cell wall. They have a wall. They have a membrane as well. They have a membrane as well. It sits out here. So if a plant cell looks something like this, and we put it in 3 different conditions, if it's in isotonic, plant cell is isotonic, then we're going to leave it alone. It's the same. No problem. If that cell is put in hyper, one thing's going to happen. And if it's in hypo, something else is going to happen. So what? And when I say hypo, I mean tonic, low solute outside. Hypertonic is high solute outside. So when could this happen to a plant? Think about the winter along a road, and you see a tree. If during the winter we put salt on the roads, as you put salt, on the road, that's going to make the water the ice, as ice and snow melts, that water is going to be salt water. Salt water is hypertonic. That's why plants have a hard time living wherever you're salting roads. It's very hard for trees to do well along the side of a road because there's a lot of salt water in the winter in places like Cleveland. Now, when we're talking about hypertonic, hypotonic, what's going to happen in this plant cell? So in a hypertonic solution, where's the water going to go? It's going to move out. It's going to move towards the hypertonic. Water always moves towards the hypertonic side, period. Or you can think of it as going to from waters high to waters low, because if this is hypertonic, that means there's a high solute or a low solvent. And the solvent is water. Hypo is in here. And that means it's low solute or high water. And remember, you got to think about this as a relative measurement. We're talking about a percentage, guys. We're talking about a pie chart. If I have a pie chart where normally, if most of this pie chart is water, and this is the solute, and this is water. If I increase the amount of water here and decrease the percentage of solute, then what we're doing is this becomes hypotonic and the other one's hypertonic. So if I have this difference where this is water, which one of these is hypertonic? Well, this is hypertonic because this has more solute compared to water than this one. Do you agree? This has a higher percentage of solute. So you have to think of this as a relative measurement. All right, so that's hypertonic. Water's going to move out. The more in hypotonic solutions I have more water relative to solute, and in hypertonic solution I have more solute relative to water. So consider that. In a hypertonic solution, then water is high inside and low outside in this condition. So water is going to move from its high to its low. Or you can just remember the simple rule: in osmosis, water always moves towards the hypertonic. That's it. So when we're talking about hypertonic solute and solvent, I forget long loss. There's so many things that are distracting. All right, hypotonic. When we put something in a hypotonic solution, which way is water going to go? Well, yeah, but what? Where does water always move? What's the rule we should memorize? What's water always going to move? Hypertonic. If this side's hypotonic, what's the other side? So this must be hyper. So that means water's going to go in. Just that easy. See. Now, if water's going in, what's going to happen to this thing? Oh, by the way, I should mention, if water's leaving, what's happening to this membrane? It's shrinking. Now, this is called plasmolized. And you can see the drawing. They draw it better in the project. What just happened there? I've never seen that happen. Have you ever seen that? It's a new function, I guess. Plasmolize, all right? All right, so in animal cells, they're called crenated. So they shrink, it's called crenated. And in plant cells, it's called plasmolized. Just one of those differences you have to know. You'll see it. It's not really that big a deal. They're both shrinking. I'm not sure if there's a difference in how they react. I've never known. They're both going to end up dying if they get too plasmolized, okay? That's in a hypertonic solution. For a plant, what that means is the plant will wilt. The plant wilts, and obviously that's not, it looks sad, but it's also dying. All right. For a human, our skin just shrinks up, shrivels. You'll see it happen to animals. You'll see easily single-celled organisms will just shrink. We could do that under a microscope. You could see them happen. I don't like doing it because I hate killing things. Let's see. Where is it? Hypertonic, hypotonic. So obviously water's going in. What's going to happen to a plant cell? So it's going to swell. So that's for an animal cell, that's awful because an animal cell can burst in something we call lysis, right? We've gone over all this already, but I'll do it again here. It'll swell up and pretty much fill the space. But why won't it lyse? It actually becomes turgid. Plants like it. We call it turgid. It's Tuger pressure as the membrane pushes up against the cell wall. Why doesn't a plant cell lyse like an animal cell? That's right, the cell wall keeps it from bursting. And so what happens to this plant is it's standing straight up. And so this plant stands straight up and it does not wilt. So if you look at a flower, let's look at a flower here. It stands straight up. And the way plants stay straight up is the water's pushing up on the sides of the cell walls and it holds it up. That's what allows the plant to hold straight up. If it loses that water pressure, it just kind of sinks down and wilts. All right, so that's plant cells, and obviously they're balanced. There's no different. They're equal concentration on both sides. There's no real net movement of water. So that's what this looks like. It's turgid and plasmolysed in plants. It's crenated and lysed in animals. Did that answer your problem with 21? Yes? They should have. I think you should be able to answer the question now. And by the way, remember the answers are in the back of this packet. Yes? If you ever have an issue. That's why I gave you the answers. All right. Now, what's the other one? Why didn't it give me that warning earlier? I don't know, but it didn't. All right, so you have this thing called the paramecium. We had one in class. We had a live one from the pond sample, the water sample I grabbed from the pond. It's a ciliate. It has a bunch of cilia sticking out. You have the drawing there. In the Paramecium are something called, they're called protease. They're protease, which means that they are eukaryotes. They do have a nuclei. In fact, they're really weird eukaryotes. They're really, really strange eukaryotes because Paramecium have more than one nuclei. They actually have a macronuclei. And they have micronuclei. They're really strange. In fact, you know, we have male and female. They have 6 different sexes. They have a type 1, 2, 3, 4, 5, 6, no, 7. 7 different sexes. You know, male and females can have babies without science, when they help from scientists. But male and males can't have babies, and females can't have babies, right? You need a male and a female. Well, Tetrahymena thermophila has 7 sexes. You get type 1 can have babies with any of the other types, but they can't have babies with type 1, 2, etc., the same thing. And so they have 7 different sexes. That's insane. And when they have sex, they exchange this micronuclei. The whole nucleus goes. They send the other. It's crazy. So that's a protease. And protease, this paramecium, as we know, spends its time in fresh water. What is fresh water to you? Is the water that comes out of the tap fresh water?
No.
Yes, it is.
But it's processed.
It is processed, but it's when they say fresh, they don't mean it's freshly made. It means there's very little salt. There is salt in all water, but it's little salt as compared to what? Well, not dirty. It's not, well, okay, dirty too, but it doesn't have to be dirty. There's something else that's not fresh water. It's called salt water. There's salt water or there's fresh water. Salt water, there's more than 2 sources. You'll find them in the ocean. Ocean is salty water. You can taste it. It'll sting your eyes if you jump in it for the first time. There's a place called, there's these Salt Lakes. Salt Lake City, Utah is a good example. There's marshes. Salt marshes also have salt water in them. The place where there's little salt freshwater lakes like our Lake Erie, rivers tend to have fresh water. And our wells tend to be fresh water. So these Paramecium live in fresh water. They have this really strange little organelle called a contractile vacuole. It tells you in the problem that this is hypotonic. Water is always going to move from hypo to what? Hyper. If outside is hypo, inside is hyper. So water is going to keep flowing in. How's this cell going to deal with that? If it's going to keep going in, it's going to plasmalize. It's not a plant cell. It's going to lyse. It's not a plant cell. So what's going to happen? Has to get the water out. It has to lessen it. That's right. Well, what's it going to use? It's going to use this little guy right here. This little guy is a vacuole. A vacuole is a space, a compartment. It fills up with water. All the water goes in, and then what does this thing do? It does what? Contracts, and that just spits the water out. Then it fills back up and contracts. It literally pumps the water out using that contractile vacuole. Like if you were on a rowboat in the ocean and it was leaking, you had a bucket, what would you do? Scoop the water out and hope that somebody saves you soon, right? Before you run out of energy. That's what this thing's doing. They pump the water out using this contractile vacuole. So they live this way every day. This thing's running 24/7, getting all the extra water out. Water's coming in. They know they can't stop it, but they can pull it out. Okay? So that is the solution to that problem and why that problem is the way it is. Are we all good? Any other questions? I had a better explanation and better drawings, but I deleted them, unfortunately. Yeah.
Can I get clogged?
Can you get what?
Can I get clogged?
Clogged? What is that?
Stop, like a stopper?
I-
Can you clog the toilet? Can that get clogged?
Oh, can this get clogged up?
Yeah.
No. Because it's osmosis allows only water to come in here. Water's the only thing that gets in here, and then just spits it out. I don't know if I guess it could if you had the right molecule, but no, I wouldn't worry about that. They're constantly doing this in nature. They live in ponds and rivers and such.
Oh, that's like an animal?
It's a single-celled organism. We call them protists. Protists can be very, very, they're eukaryotes. They have a nucleus. But they can have the algae can be protists. They have paramecium, all kinds of ciliates. Amoebas are protists. They do all kinds of crazy things. These guys eat bacteria. They live in rivers and lakes, and they eat bacteria.
And can you actually see that?
Yeah, I showed it to you. We had it in pond water. If you didn't see it, I'm sorry.
You can see it with your naked eye?
With a microscope. They're small. They're tiny little things. But they'll sit around. They'll swim around. You can watch them eat. They just go like this. They're good for us. They eat the bacteria. We don't want bacteria, right? Amoeba do too, but amoeba can also cause disease. So, all right, are we good? Any other questions on this packet? Please be ready for tomorrow's test, especially if you've been laying your head down. Certain people we won't mention, gentlemen. They're so asleep. Hello. You slept through a third of the class. I hope you are ready for tomorrow's exam. Right, gentlemen? All right, good luck.